Right, these are going to be your lecture notes for this week. Please title them Cell Reproduction. I would like these lecture notes completed before you come to class on Thursday. I'll be checking them off in class on Thursday for credit. It's important that you do these notes before class on Thursday, since you're going to need to know some of these facts to do the activity of what we're going to do in class on Thursday. All right, so we're going to be looking at cell reproduction for the next few weeks. This will take us through to our final. Cell reproduction is looking at how do cells make new cells? How do they copy their DNA? How do they replicate? How do they divide? And why? Why do they do this? So you need to think back to when we first were learning about cells. We learned that there were two different types of cells. There were prokaryotic cells, which include bacteria, and then there were eukaryotic cells, which were the plant and animal cells. Prokaryotic cells, if you remember, didn't have a nucleus. They actually had just a single long chromosome inside of them that was floating around. They had the cilia, the hair around the outside of them. They had a flagella, the tail that would help them move. And the way that they reproduce is they reproduce asexually through a method called binary fission. And what binary fission, the steps of binary fission are, first, the cell will increase the cell mass slightly, meaning the cell will get larger. And then the DNA and the cell components inside will replicate, meaning that they copy themselves. And then the cell will divide in half and break off, and you'll have two daughter cells here. So you'll end up with one on each side. What we're going to be looking at for this next few weeks, well, let's back up. Actually, the most common prokaryotic cell that does this binary fission is a bacteria cell, and specifically the one that you might be most familiar with is the one that's been in the news lately with Chipotle, and that is the E. coli cell. And what E. coli does is it just copies itself and breaks in half, and now you have two new daughter cells, and this bacteria cell divides very quickly, and that's why you have a problem with people getting sick, and it's a dangerous bacteria to have as well. So, but let's look at the eukaryotic cells. So these are the ones we're going to really be looking at in the next few weeks. They are the cells that we've already been studying a lot. They have the nucleus, they have all the organelles inside that we've been learning about, the mitochondria, the lysosomes, the ribosomes. And these cells can reproduce themselves asexually through a process called mitosis. Mitosis is also called cell division, which means the division of cells, and it has some very distinct phases or stages. So most of the time, a cell is in a state of what's called interphase, meaning it's resting, it's asleep. It's just doing what the cell's normal cell activity is. This will be the longest part of the cell's life. The cell spends most of their time in interphase, and during this time, the cell is just doing what its job is. The chromosomes are not visible during interphase. But cell metabolism is occurring, photosynthesis, cellular respiration is going on, and this is where the cell is going to be right before it decides to start mitosis, meaning right before it starts to decide to replicate and make new cells. So here's a picture of some cells that are in interphase. You can see the nucleus here, but you really can't see any chromosomes. You can't see anything inside the nucleus. These cells are just in their resting phase. And then what you're going to do is you're going to start the process of mitosis. Okay? So mitosis, well, here's a picture of a cell in interphase. You have the nucleus, you have these chromosomes inside that aren't really distinct, and you have these things over here called centrioles that are inside of the cell, floating around in the cytoplasm. But they don't really have a job yet to do. Okay, you'll see what their job becomes once mitosis starts. All right, so the cell is going to start its replication now. It's going to make a new cell, and it's going to go through 4 stages of mitosis. It's going to go through a prophase, metaphase, anaphase, and telophase. And some people memorize it as PMAT. You can make a mnemonic device like Paula must answer telephone. I don't know. Whatever you decide you want that to be, but it's PMAT. So let's look at the first stage that they're going to go through, which is prophase. This is a picture of cells going through all different stages of mitosis. And once we've done this lecture, you'll be able to specifically look at this one and go, okay, this one's in interphase, this one's in metaphase, this one's in prophase. So let's look first at what happens when they start in prophase. So in prophase, the DNA inside the nucleus is going to start to coil. It's going to become visible. The nuclear membrane is going to start to disappear. And those centrioles, the things that we saw that were floating around in here, are going to start to migrate to the poles, meaning that one is going to come up here, one pair is here, and one pair is going to come here. Okay, so this group of centrioles right here are going to start to come up here, and this group of centrioles right here are going to start to come down here. And then you're going to see what they call these spindles start to form. So here we go. This is a picture of prophase. You can see the chromosomes here. You can see that these centrioles are starting to move. One's coming up here, one's coming down here. And they're also starting to get these things on them called spindle fibers. They're starting to grow these little spindle fibers, and you'll see why they need to have those. Okay. And when we're looking at a picture of prophase under a microscope, you're going to see something that looks like this. And this is just the DNA, the chromosomes, just uncoiling and starting to get really tight and visible so that you can see it. So we're talking about chromosomes, and this is a picture of a chromosome. This thing in the middle is called a centromere. This thing that's going to connect it are called the kinetochore. And you can see this kinetochore starting to get these little things on it called spindle fibers as well. This is one half of your chromosome, and this is your other half of your chromosome. And in humans, we have 22 chromosomes. We have a 23rd one, which will distinguish if you're a male or a female, your X and your Y chromosome. But if you look carefully, each of these chromosomes represents something, and there is a pair for each one. Okay. So we went through prophase, and now we're going to look at metaphase. And during metaphase, these spindle fibers are going to start to come down here from these centromeres, centrioles, and they're going to attach to the chromosomes. These chromosomes are going to line up on the equator in a straight line with half of this chromosome on this side and half of this chromosome on this side, and half and half. So the first thing that's going to happen is that these spindle fibers are going to come down from the centrioles and attach to the chromosomes. So these come down and attach to the chromosomes. Then the next thing that's going to happen is the cell is going to start preparing to separate the chromosomes. So all of these chromosomes are going to line up right in the middle. Okay. Right after, well, let's look at metaphase. So you can kind of see this one, how they're lining up all in the middle. This cell right here is in metaphase. You can even see, if you look really, really close, these like spindle fibers that are attaching here. Okay, so prophase, metaphase. The last one is the third one, sorry, is the A one, which is the anaphase. And during anaphase, these centrioles with the spindle fibers are going to start to pull back. It's almost like they're going fishing, and they're going to start to reel back in their spindle fibers, and they're going to start to pull them in and pull them in. And these chromosomes right here are going to split. So half of the chromosomes are going to go on this side of the cell, and half of the chromosomes are going to go on this side of the cell. And each of these halves of chromosomes are called daughter chromosomes. Okay, so the cell chromosomes are going to separate. The spindle fibers are going to shorten so that these chromosomes will pull to each of the poles. Okay, you're going to draw a picture on your notes of how these spindle fibers attach to the chromosomes. And you might want to go back and look at the one slide that we had that was the picture of the chromosome and see how this is where they connect right here on this centromere. Okay. Then we're going to go into the last stage of mitosis, which is telophase. And this is when the chromosomes are going to separate completely. You're going to get your nucleus back formed, and the cell is going to do something right here, which is called cleavage, which means it's going to push in here and push in here, and it's going to actually pinch off the cell so that you have two separate cells. You can see these centrioles up here. The spindle fibers have become detached from the chromosomes. You have this daughter set of chromosomes here and this daughter set of chromosomes here. And then inside the chromosomes, they're going to start to uncoil again and just become a long chain, not specifically a chromosome. So when you're looking at them, where are these two sets of chromosomes located during telophase? They're on the poles, they're on each end, and they're actually starting to get formed back into the nucleus membrane. Okay, so the two things that reform during this time are the nucleus and the nucleolus. So now the chromosomes are not visible anymore. The last thing that's going to happen, it's not really a phase, it's just something that happens at the end of telophase, is something called cytokinesis, meaning that you're going to form these two identical daughter cells, and that this part right here is going to actually break and float, and they're going to be in two separate pieces. So here is a picture of a cell in telophase. You can see that this is the cell here. The chromosomes have gone to each side, and now it's starting to break off into the middle here, but it hasn't completely pinched off yet. It's still attached. And even on this one, if you look really carefully, you can even see the spindle fibers on this. So then the last thing that's going to happen is cytokinesis. It's going to break off, and then you're going to be left with two different cells. This is going to happen now. This is going to rest maybe for like another 80 minutes, and then it can replicate again. Some cells can replicate very quickly. Like if you have malignant cancer cells, sometimes they can replicate and reproduce so fast that you can't even keep up with trying to control them. And other cells reproduce much more slowly. So I'm going to show you this video here of the steps of mitosis, and you can take some notes on it, but be prepared when you come to class on Thursday to know the basic steps of mitosis, their names, and the key points of what happens during those stages.
Cell division is required for an organism to grow, mature, and maintain tissues. During the mitotic phase, a cell will undergo mitosis to form two new nuclei, and then divide to form two new individual cells during cytokinesis. Mitosis is the process of dividing the duplicated DNA of a cell into two new nuclei. Mitosis is split into distinct stages. The first stage is prophase. The DNA condenses, organizes, and the classic chromosome structure appears. Next comes prometaphase, where microtubules attach to the chromosomes. This step is followed by metaphase, where the chromosomes align. Metaphase is followed by anaphase, where the chromosomes separate. Finally, during telophase, nuclear membranes reappear around the two sets of chromosomes. Mitosis is now complete. After mitosis, two new cells are formed by a process called cytokinesis. Mitosis is only one part of what is called the cell cycle. For many eukaryotic cells, a cell is duplicated every 24 hours. Most of the life of a cell is spent in interphase. Interphase consists of three stages called G1, S, and G2. G1, or gap 1, is the first growth stage of interphase. In G1, the cell grows to nearly its full size and performs many of its specific biochemical functions that aid the organism. Next is the S, or synthesis phase. This is an important stage because it is during the S phase that DNA in the nucleus is replicated. The cell next enters another growth stage called G2, or gap 2. It is during G2 that the cell finishes growing. Once the cell has duplicated DNA in the nucleus and two centrosomes have appeared in the cytoplasm, mitosis can begin. For a typical eukaryotic cell, this will last about 80 minutes. During the first stage of mitosis, called prophase, we first see the classic chromosome structure. This occurs through a condensation process. At the same time, protein strands, called microtubules, appear from the centrosomes in animals. Finally, a structure found within the nucleus, the nucleolus, disappears. Next, prometaphase begins when the nuclear membrane is broken down. At the same time, microtubule strands, or spindle fibers, are growing from the centrosomes. These strands attach to a protein structure called the kinetochore. One kinetochore is attached to the centromere of each sister chromatid. Next comes metaphase. During this stage, the sister chromatids align along the center of the cell so that both chromatids face toward opposite poles of the cell. Now the sister chromatids are ready to be separated. This occurs during anaphase through a shortening of the microtubules attached to the kinetochores. Additionally, the poles of the cell move farther apart, causing increased separation of sister chromatids. At the end of anaphase, the sister chromatids have moved to the two ends of the cell. Telophase is the final stage of mitosis. It is here the components of the new cells begin to appear. At this point, the spindle fibers are broken up. A new nuclear membrane surrounds the chromosomes at the end of each cell, and the chromosomes uncoil and return to an uncondensed state. Mitosis is now complete. The formation of two cells is all that remains. Following mitosis, the cell undergoes a process called cytokinesis. First, the cell is compressed by a contractile ring that divides the cell in nearly equal halves. By now, the organelles in the cell have been replicated and are now divided between the two halves of the cell. This includes mitochondria, Golgi bodies, and the rough ER. Plant cells also have chloroplasts. Once split, the two new cells are now fully in the G1 stage of interphase and ready again to begin their growth. Let's watch the process one more time. Mitosis begins with prophase. Notice the DNA condensing into chromosomes during this stage. Microtubules appear during prometaphase, and the nuclear membrane breaks down. Metaphase occurs when the chromosomes are aligned at the center of the cell. During anaphase, the chromosomes are moving apart. The telophase stage is marked by the appearance of new nuclear membranes. This is the end of mitosis. Finally, the splitting of the cell occurs during cytokinesis. The two new cells are now ready to grow and perform their specialized functions.
Okay, so that's the end of that. I will see you in class on Thursday. Please make sure that you either submit your notes through Schoology before class on Thursday or have them in your notebook. What I would prefer is I would prefer them either in your notebook or you print them out and put them in your notebook once you submit them on Schoology. I'd like all of your notes kept in the notebook in one central place. All right, see you Thursday.